10 years, we actually see if we can take information out of RNA and see if we can get it to write itself into DNA.
What's RNA?
Well, so that's ribonucleic acid. That's the little information packets that actually inform cells how to make proteins.
Okay.
We're going to see if we can get those to actually modify the actual genes, the actual information of how the genetic systems of a cell work. And we're going to see if we can get RNA to write itself into DNA and modify an organism. And so for 10 years, the National Science Foundation funded a project called Darwinian Chemical Systems. And 10 years into it, a company called Flagship Ventures came along and started a little company that no one's ever heard of called Moderna.
Okay, that's where Moderna come from.
And people sit there going, oh, but they said RNA wouldn't affect our genes. They said it wouldn't write into the human genome. It wouldn't change what people are. It wouldn't alter what people are, except for the unfortunate reality, which is the reason why the company started, is they succeeded. They succeeded in getting RNA to change and modify the genetics of living systems. And the first clinical trial that was done on that technology was called the COVID vaccine.
What year was this?
Now, during the pandemic, 2020.
Okay.
Here's the interesting thing about that statement.
Yep.
And it's super interesting. The head of the Institutional Review Board of one of the largest universities in America on the West Coast. I'm not at liberty to divulge which university, but I will tell you, it is one of the largest. It might have the name of a state involved in it, but I can't say what it is. But let's just say, very large university, might have the name of the state in it. You get where I'm going. The head of the Institutional Review Board, who is the scientific ethic body that's supposed to review studies, called me to tell me that when she asked for the data, which were the animal studies to prove that there had been any safety study on the Moderna injection at all, she was told, and I quote, this was a straight-to-humans protocol.
There was no animal testing.
No animal testing.
At all.
So how do you actually come up with safe and effective? How do you come up with informed consent? How do you come up with anything when the very basis of scientific research, which says that what we do is we start out at a petri dish, then we go from a petri dish to a cell line, then we go from a cell line to different kind of animals. We do a whole bunch of things to say, oh my gosh, if we see liver cells die, or if we see epithelial cells die, or we see anything else mutate, we actually say, oh, that's not safe. But we do that in systems long before we get to humans. And when asked for the safety data from the animal studies, their response to the head of the Institutional Review Board at this major university on the West Coast of the United States was, this is a straight-to-humans protocol. Now, what does that mean? That means that we took not only a risk, but we knew that this technology was going to alter what it literally means to be human. We were going to do what we've done to seed crops. We were going to do what we've done with hybrid vegetables for a long time. We were going to do that, but we were going to do it with humans with not a single piece of safety data to prove that it worked.
Why? Why wouldn't they have done that?
To kill people.
To
kill people. We can dance around this thing for a million years, or we can actually get to the point.